Good evening, everyone. Thank you for joining us tonight to hear Dr. MAdreani's talk. I'm Lisa Collins, and I'm the president of Scas. We're really excited to have you all join us again for tonight's event. This is our last event for this inaugural series, spotlighting our board members and their research and contributions to science. We hope these events increase your connection to Scas and show you the value in continuing to support Scas and our mission to support the science and the scientists working in Southern California. For our guests, if you enjoy this talk, we encourage you to join Scas. Your membership supports scientists and students in Southern California. We hope to see you all at this year's annual meeting, which will be held in a similar virtual format. The annual meeting will be on Friday, May 7th from 8 a.m. to 5 p.m. Information on the annual meeting, including registration details, can be found on the Scas website, scas.nhm.org. Registration prices start at just $15 for student members. I'm pleased to introduce Dr. Jason Hui, an alumnus of the Scas Research Training Program from 1991 to 1992. He earned his BS and MS in electrical engineering and his PhD in mechanical engineering. All those degrees were earned at that lovely university down the street, UCLA. Dr. Hui is currently a senior staff program engineering manager at Elbit Systems of America, based in New Hampshire. We're excited to have him join us tonight for this talk. Thank you for being here, Dr. Hui.
Thank you, Lisa. And I'm happy to be joining you here from New Hampshire, which is 3 hours ahead of LA time. So it's my pleasure to introduce our speaker, Dr. MAdreani. Dr. Adreani is a research biologist at Cal State Northridge and an adjunct professor at Occidental College. She received her Bachelor of Science in Aquatic Ecology at UC Santa Barbara, Master of Science in Biology at Cal State Northridge, and a PhD in Biological Science with focus on ecology and evolution at the Florida State University. Dr. Adreani has broad interests in the evolution of mating systems and behavioral ecology, and her primary research is focused on investigating mating behavior, alternative mating tactics, and fertilization dynamics in marine fishes. Tonight, she will present her research on the reproductive ecology of sex-changing fishes in the Southern California Bight. Before I turn it over to Dr. Adreani, for all our attendees, feel free to type in your questions in the Q&A box, which you can find the icon at the bottom of your screen, and we'll get to those after the presentation. With that, Dr. Adreani, the floor is yours.
Thank you, Jason. Is it working? Oh, there we go. Okay. You guys see that?
Yes.
Thank you. Okay. All right. Hi, everyone. Thanks for coming and joining in on this talk with Scas. I'm happy to be a board member of this organization and have really enjoyed my involvement with them over the years. So happy to share a little bit of my work for everyone. And I'm going to just give you kind of a little blip, a little tidbit about some of the work that I've been doing over the last, well, several years, including a little bit from my master's work at Northridge, and then from the last several years working here as a research biologist in LA. Excuse me. Okay, so one of the aspects, one of the big questions that I've often asked about over the years of studying fishes are what are the mating behaviors and mating systems of Southern California fishes? And course, that's a broad question, and I'm going to give you guys a few examples of that, ones that I've worked on specifically, and certainly a lot of people have worked on this over the years. I'll draw in some examples from elsewhere, but I'm going to focus in on kind of 3 case studies for the sake of this talk. And I guess it's important to start out with, why do we care about this? I obviously have an interest in it, and just found it fascinating that fishes kind of do everything with respect to their reproductive biology and their reproductive behavior. But why do we care as a public? Some of that goes back to there being somewhat limited knowledge of mating systems in temperate habitats. I got my start working in temperate habitats, meaning somewhat colder habitats like the coast of California, as opposed to tropical habitats where a lot of work has been done. And there are reasons for that. It's a little bit easier to work in tropical habitats because of better conditions, clearer water where you can actually observe fishes, and being one of them. And some of these data can provide additional data for fisheries management that we didn't have before. And that's particularly important with respect to sex-changing fishes. And I'll get into that in a moment. But a lot of our popular fishery species are sex changers. Many of you may enjoy things like grouper and other fishes like that. Those are sex-changing fishes and will relate to some of the species I'll talk about this evening. And so knowing more about how they reproduce, when they reproduce, how often they reproduce, can help to aid in management of those species so that we have them around for consumption into the future. But I'll add the caveat that studying highly mobile animals can be really difficult. So I'm going to give you 2 examples of very mobile species, one of which is a fishery species. And then I'll give you a little kind of a little short story about a species that we worked on that is less mobile and more manageable to work with. And we tried to mimic some of the aspects of the mobile species to see if we could find out a little bit more about sex-changing species in a fishery situation. All right, so let's get into fish spawning. So there are lots of different ways that fish spawn. I'm going to give you a couple of examples, and there are many more. As I said before, they kind of do it all. So one way is to pair spawn. We have 2 individuals releasing sperm and egg. Okay, and here's an example of 2 hamlets that are exchanging eggs and sperm in sort of a cupped behavior. And then you have the other end of the spectrum where you have group spawning, where you probably have 1, maybe more, but likely 1 or 2 females and a huge number of males, hence the cloud of sperm in the water, where they all sort of gather together in these large groups or aggregations for spawning events. These are both tropical species. But now let's take you into the kelp habitat, which is where I've done most of my work. I've done a little bit of work in the tropics, but for the sake of this talk, given that we're centered here in Southern California, I'm going to talk about kelp forest habitat. And here it is from the surface when one has a nice year with lots of kelp, which hasn't happened in a couple of years. This is one of the reefs where I've done some extensive work over the years off of San Clemente, California. Okay, so my first species that I'm going to talk about is the California sheephead, Semicostophus pulcher. This is a protogynous meaning female to male sex changer. Okay. There are only secondary males, and I'll get a little more into the definition of that, but essentially that means that all of the males once matured as females. So they all went through a sex change pathway. I'm going to give you an example next of a species that can have both primary and secondary males. So I'll make that distinction in a moment. In this mating system and their behavior, one male will mate with multiple females sequentially in the course of an evening. So when I've observed them spawning, a male will start to become a bit territorial in a particular area of the kelp, and females will start to gather. Both males and females can become aggressive with each other, competing for access to both males and females. So the females will actually fight for access to the male. The males will fight for access to all of the females. So one male mates with multiple females in an evening. There we go. Sorry, my slide wasn't changing. All right, so again, just to reiterate, these are female to male sex changers and only secondary males. Why do this? Okay, why change sex? Well, for a lot of fish species, and again, this is relatively common amongst fishes, becoming larger and territorial can actually lead to greater reproductive success. So I described briefly what that mating system looks like. You have one male that dominates, kind of fights off the smaller males, and then mates with multiple females in an evening. The females are only probably mating once in that setting, okay? So if you can become large and gain access, and the larger male maybe leaves or dies, and if you're the largest, most dominant female, this is why they fight amongst themselves, then they're the one that's likely to change sex and become a male, then be able to mate multiple times in an evening. And so ultimately, it's about increasing your reproductive success. So why is it important to study sheephead reproductive ecology? This is a popular sport fish. So some of you may be familiar with it if you fish or just have an interest in coastal ecology at all. There is a live aquarium trade for them at smaller sizes. So they're important both for aquarium trade and also for sport fishing. They're target species often for the formation and monitoring of marine reserves. Okay, so again, just to reiterate, sequential hermaphroditism, female to male. And again, this is a popular sport fish. So when a large male, which is the target, right? When you're going out to fish, you want the largest individual. Those are going to be male, okay? So what does that do to the population if you're largely taking out males, which are already underrepresented in the population? Heavily skewed toward females because only the largest, more dominant ones become male. And again, here's just some photos. It came out a little grainy, sorry. Of in the upper left, you have two female sheephead jawing at each other, which is kind of aggressive, and then two males in the lower right. So they do fight underwater. They don't usually come to blows, but they will definitely display and seek dominance. Here's an example Again, a little bit grainy because they spawn at dusk. And this is a video that's, oh shoot, okay. The video didn't connect. It's giving me a little warning. All right, so here is an example of a spawning bout. There are a couple other species. Lower right, there's a kelp bass down there. And in the distance, there are a couple other sheephead kind of hanging out. But this was, you can see the large male in the center there with a female. And I believe this was taken just as they were, they had just spawned and then they were releasing back down into the water column, where he will then kind of sidle up to another female and spawn again a little bit later. And I did include a little diagram, thank you to Larry Allen, which kind of highlights the spawning pattern, the behavioral pattern, where a large female will kind of swim around, dominate an area, kind of set up a territory on a particular evening, and then start to court females in the area. They are kind of drawn in, and there will be multiple females, 5 to 7, for example, around the area. He will then do this behavior with each of them sequentially. So it's a pair spawning, as I mentioned before, but with multiple females in a particular evening. The other aspect of my research includes looking at their physiology and the internal part. And so I described some of the behavior that I've witnessed while diving and watching them. And the other thing that we are able to do with them is look at their gonads and get a lot more detail about how many eggs are actually released when they spawn, and also how often they spawn throughout a season. We do that by dissecting them. Obviously, that means we have to catch them and kill them, which isn't great all the time, but we do. And sorry. And we can remove their ovaries. And so that's what's happening on the upper left. The middle photo is a close-up of an ovary where you can kind of see, hopefully you can make out that there are some clear spots that look kind of watery, little water droplets, which indicates that they're hydrated, which is a sign that they're about to spawn. On the far right is an example of what we see when we dissect out a portion of the ovary and look for hydrated eggs. And hydrated eggs are a really good proxy for how many eggs they would have spawned that particular day. Okay, so we can get a batch, what's called a batch fecundity, meaning that they release little blips of eggs during each spawning event. So those slightly larger and clearer eggs in that little bundle are the ones that would have been released that day. So what does that look like? I'm showing you one tiny little droplet in that photo, but what that actually amounts to are tens of thousands of eggs per spawning bout, okay? And upwards of 50,000. So they go for it and try to release as many as they can, because a lot of them aren't going to make it. And this is one graph just showing over the course of about 9 years, the fluctuations in that year to year. And we do think, in this case, I was trying to look at it with trends of warm water years to see if there were differences, and it does appear that in warm water years, they release more. Their fecundity is higher, their batch fecundity. And that's something we can talk about more. If anyone's interested, I can talk about that more. But I wanted to show you one example of what comes out of some of these data. We can also look at things like sex ratio. So in sex-changing species, typically for protogynous, meaning female to male sex changers, the population is skewed heavily toward female. There are many more females in the population, a few males. Okay. Excuse me. And so again, this is trending over years. And does that shift with other environmental factors, such as, again, temperature, maybe food availability, heavy fishing, areas of heavy fishing where there's a lot of removal of males. Okay, next one is not a fishery species. So this will be another kind of a shift, another kelp forest fish that's very common in our waters here in Southern California, but not one that is targeted by fisheries. It's a little bit smaller and not of great interest to people that eat fish. And so this is one that I worked on during my master's thesis and was interested in describing what their reproductive patterns were, both behaviorally and physiologically. So they are a diandric protogynous hermaphrodite. Okay, so what does that mean? Well, it means that the pathway to becoming male, there are two of them. There are two pathways. So some individuals will develop and mature as males. So going from a juvenile to a male phase directly. Some will develop and mature as females, but later in life become male. Okay, so that is what's called a secondary male. So they've mature first as a female, like the sheephead, then change sex. But in this case, in this species, they can actually develop directly into males. They also are really interesting because they exhibit both group and pair spawning. And it seems to be density dependent. So in smaller densities, they seem to pair spawn, and in high density, they group spawn. And I'll show you some examples of that. So here are the different phases in this species. Here's a juvenile in the upper left, what's called an initial phase. An initial phase could be male or female. And then a terminal phase. And terminal phase is always a male. And you can see that there is quite a distinct color pattern and a bit of a shape difference to them as well. But in particular, that dark band behind the pectoral fin and a little bit brighter coloration. When I was studying them, I wanted to describe what that sexual pattern was. These are fish that live about 7 to 8 years, but the bulk of the ones that in the populations I was studying at the time were in the kind of 1 to 4 range. And so that's what's indicated here. And the thing to note with this graph is simply that males and females are present at all size classes, but secondary males are only present after the first year. So they've already matured as females and then can transition to males. And here is another one of those diagrams indicating their reproductive pattern, their spawning pattern. In the pair spawning pattern, they will court. So it's a little more energetically costly for them. They will court females, not unlike what the sheephead do, in that they, will kind of do a little bit of dancing, so to speak. So that's what's indicated by this sort of zigzag pattern that you see. with the colorful male there, kind of zigzagging in a circular pattern, and then kind of sidling up to the female, and then they go up into what's called a spawning rush and release egg and sperm. So that's one variation in their behavior. The other option that is more common is a group spawn. This is typically done with the smaller males and female. So it's a little bit more like what I showed you with the snapper species, where you have one female being kind of chased by a bunch of males. And then they kind of go up into a spawning rush with lots of males and probably one, maybe two females in the mix, but usually just one. That obviously has implications in that there's multiple paternity. Those eggs are probably getting fertilized by more than one of them, but they're competing for access to that female still. And so in all likelihood, the one that's closest to her is going to fertilize more of her eggs. So there is competition there. My last example is one that we actually, I was talking a lot about mating behavior and observing them and observing their spawning patterns and their spawning behavior. And one of the ways to get at this idea of large males being removed from the population, it's really difficult. As I mentioned before, it's really difficult to work with large, highly mobile species. So one way to get around that is to work with a tiny species that's also a sex changer that can mimic some of the aspects of those larger species. So a group of us went about this using this species as a model species. And I know you can't tell from the scale, but this little goby is probably about 3 inches long. So they're little. And we can use them as a model to, again, mimic what happens in a population when you remove larger individuals, meaning the males, from the population. Do you see a crash? Do you see compensation by the females? Is there a greater chance for them to change sex and compensate for the lack of males? Or do they crash, reproductively speaking? So this work, excuse me, was done at the lovely Catalina Island off the coast of Southern California. And right here in this cove at Wrigley Marine Science Institute. Excuse me, throat's a little dry. So we worked out here for 3 summers and set up multiple experiments that involved putting these animals in little cages. So part of the fun of doing ecology is that you get to be creative about designing experiments and then building them. And doing them underwater is a whole nother level of frustration, but also kind of creativity. So we set up an experiment right here, kind of under, just next to where those boats are in this area over here, and set up a little goby habitat, little goby condos. And so this is the build out here. We're building these cages, which I'll show you what they look like underwater in a moment. But I show this just to indicate that there's a lot of work that goes behind these seemingly very simple experiments. There's a whole lot of stuff on land that has to happen and lots of trips to Home Depot to get all the materials. And sometimes we get a little wrapped up in it, like my colleague here, caught up in all of this hardwire mesh that we built these cages out of. All right, and this is the end product here. These nice little cages that were roughly 6 by 6 feet, where we were excluding predators so that we could kind of see what would happen with these gobies under different treatment conditions. We also used terracotta saucers. If you were wondering, those looked kind of familiar. Yes, they are your typical thing that's under your pots and plants outside. And they will actually lay their eggs in them. So these are nesting species. They lay their eggs in little nests. Normally those would be maybe under a rock, a little ledge of a rock. And so they're pretty happy to use these terracotta tiles to lay their eggs. And that's fortunate for us because it makes it easy to monitor them and actually count how many eggs they lay. And there were a lot of different aspects to this experiment. I'm only going to highlight 1 or 2 of them since we're talking about reproduction and reproductive ecology. But this is what one of the egg mats look like on the lower right here. And to give you a scale, there's a little ruler there. So they're fairly small. So we had hypothesized with some modeling that that the proportion that of transitioned, again, sex changed, right? Portion of female sex changed would increase with a lack of males. So more females changed sex when the proportion of males was low. Okay? And no sex change occurred when there were more than 40% males. So having males in the population basically dampened the rate of sex change by females, which makes sense for a sex changing population. But then what happens when you take males out of the population? And that's one of the things we wanted to address and use this species as a mimic for that, for what would happen, for example, in a species like sheephead that is targeted for their large males. Can females compensate for that by changing sex earlier or quicker? Or does the population just grow, or do they not reproduce at all? So essentially, here's what we found, and I'll give you a little summary of this in the next slide. But we found that the lowest reproduction occurred when large gobies were removed. Okay, so that's not shocking. It's kind of what we expected to find. We would expect that large gobies, especially the large males, were responsible for a lot of the reproduction happening in a group. Okay, and when they're removed, that reproduction would go down, and that is essentially what we found. And keep in mind that we did various iterations of numbers of removals, including some removals where they were random, where we took out some large and some small, and then some where we only took out large individuals to mimic something like what might happen in a fishing situation. And while I'm not going to show you all the graphs from all of these different aspects of this experiment, this is a summary of what we found. So sex change was 76% more higher when 76% higher when large individuals were removed at high intensity, so more frequently. And their growth actually, and I didn't share that in the graphs, but their growth was faster when those large individuals were removed. And so that indicates that there is some compensation when those large individuals are removed, that perhaps the smaller ones are trying to grow faster and then, will change sex. Perhaps even at younger ages, though we didn't do the aging aspect of it. And reproduction was much lower when the large individuals were removed, regardless of the intensity of that removal, how many we removed. So those are just a couple of examples of reproductive ecology in this area. And I hope it gives you some sense of what kind of work we're doing in this area. But of course, this is all a large group effort, and there's lots more to talk about and lots more to study. And I'm happy to take any questions or comments or discuss things further. But I'll wrap up there and just say thank you for attending.
Thank you, Dr. Adriani.
Thank you.
So I don't see any questions in the Q&A window, but I guess I can start it off with a question based on what I just heard. So I guess I'm not too familiar with fish and spawning, but I guess do you have any I guess information on what it would be like if given that we've been experiencing climate change, how does that affect what you're seeing with the spawning of these various fish and the examples that you provided?
How would climate change affect them?
I mean, are you seeing some effects from the experiments that you've been doing and how that is having an effect?
Yes and no. So despite some of the work going on over years, it's really hard to track unless you get even more data for things like looking at the details of climate change. We do have some data. I shared a little bit where we see a trend with spawning with respect to warm water years. There's a student in my lab that's working on looking at kind of the warm water blob years with El Nino and some of those events, seeing how they affect reproduction and other aspects as well. Some of that, of course, ties into food availability as well and kelp growth. So there's lots of factors there. But I don't have necessarily an overall picture for that just yet, based on my work.
Okay, great.
Yeah, but thanks. Yeah, it's good to be thinking about it, though, obviously.
Okay, so there are some questions. So the first question from one of our attendees is, are you able to identify the sex of a sheephead by its pigmentation?
That's a complicated one. Typically, yes. The males, an older and mature male has a very distinct look. When they're in transition, it can be really difficult. You might think some of them look like female, but then we dissect them out and, oh, no, it wasn't. It was just, it may have been about to transition. And so it's a little bit vague. You can tell when they're kind of older, more established males have, yes, they look quite different. But when they're kind of in between, it can be difficult to tell just by looking at them.
Okay. Another question is, other than the speed and mobility of the fish, what was another difficult aspect of studying these fish?
Well, Southern California water conditions is a big one. I mentioned that there are a lot more studies in the tropics, and a lot of that has to do with water clarity. Southern California is very hit or miss. You can have really beautiful conditions, clear, crystal clear, water, and then complete murk the next day even. So doing behavioral studies can be really tricky, which is where it's nice to be able to have the fallback of doing some of these other studies where we're actually catching them and looking directly at their gonads. So it's a good proxy when you can't always see what they're doing.
Okay. Next question is any gene similar to SRY involved in the sex determination or sex change?
I don't know. What's SRI?
SRY.
SRY. Yeah, I don't know.
Okay.
John, can you clarify that?
The question is it's from John Roberts.
Yeah.
I'm not sure. Maybe that can be taken offline. So
Yeah.
another one is how do these fish change sex? How do males and females differ genetically?
So fish do not, many fish species don't have determinate sex chromosomes, which is why so many of them don't or can change sex somewhat easily. And there are also a lot of, I didn't get into them, but actually the species I studied for my dissertation was a simultaneous hermaphrodite, meaning that they have both male and female gonad tissue. So they don't have determinate sex chromosomes like many mammals do. So it's really just an influx of hormones that shift, and it varies a lot amongst fish species, how long it takes to do that. But with the gobies, for example, it can be roughly a week to change sex. There are some that even can change back. So somewhat more flexible than I think we're used to thinking about sex change.
Okay. Next question is, when the sheephead are in the transition of changing to male, would they have both sperm and egg or just one?
Good question. Yeah. So when they're transitioning, the gonad will actually, so when occasionally we catch transitional individuals, and essentially their gonad just shrinks down to like a string, and then I don't know, for lack of a better word, kind of rebuilds going into the male gonad testes. So
okay.
it shifts. So they don't necessarily have both, I guess. In some cases, the fish will have indications of both when they're in their early, like before they fully mature. And so, it can go either way depending on hormonal influx.
Okay. Another question is, can you go into more detail about the egg batch size during warmer water years? Is the fish being stressed and in turn producing more eggs?
I actually think it might be the opposite in that that species is a wrasse, and wrasses have evolved primarily in tropical waters. We have 3 wrasse species here in Southern California that have established here, but many more species are in tropical waters. So I think they like it when it's warm. So I actually think that they are more likely to spawn. It also probably has to do with food supply throughout the rest of the year. So it's kind of a full, they don't just decide one day to spawn more because it happens to be a warm day. But if they're kind of happy and fat and healthy, they will likely spawn more that year.
Okay. Another question is, reproductive output is likely affected by habitat quality. Do you think sex change rates or female to male ratios is also influenced by the quality of reef habitat?
Yes, I would think so. In particular, the reef habitat, as I mentioned before, relating to food supply, these are benthic feeders, all 3 species actually that I mentioned. And so they are heavily dependent on the quality of the reef habitat for sure. Having both habitat when I was observing them spawn, for example, sheephead were using kind of the edges of the kelp forest to spawn. In the last few years, and a couple of years ago, actually tried to go see them spawn and didn't, and it was a year that there wasn't a lot of kelp. And so they very much are dependent on habitat for their mating behaviors, as well as food supply and having like healthy food available to them. So I think both of those tie in to habitat quality and then ultimately in reproductive output.
Okay.
Oh, I guess I didn't answer the part about sex change though. I'm not sure about that one. I have to think about that, whether the habitat necessarily would be an indicator of higher rates of sex change. So we can follow up on that maybe after.
Okay. All right. Another question is, can you discuss the RAS family as a whole group and what is more typical, primary versus secondary male strategy?
Yeah, so I think secondary male is, I think, more common amongst RASes. The diandric protogeny is a little bit less common, though not unheard of. It is the only one that we have here that does that, which is the rock grass. But in the tropics, I don't know what the proportion is, but I think secondary is the more common. So to mature as a female and then later become male. So secondary male.
Okay.
Julie, you can ask me more about that. If I didn't quite answer that, feel free to follow up.
Okay. Circling back on
the mammals. Okay.
Yeah.
I see it now. So yeah. Did I get to that with respect to that they don't have the sex determinant genes like mammals do? I don't know if that.
Yeah, so the SRY is actually the.
Now that he said it, I was like,
gene,
oh, I
yeah,
did
determine sex in mammals. Yeah.
Right.
Yeah. Okay. Another question is there a significant difference in reproductive success, reproduction patterns between primary and secondary males mating with females?
That is an excellent question that I do not know the answer to. Because as I said, we don't have a lot of data on that. Certainly not here in the temperate reefs. So for rockgrass, I don't have a good answer for that. My guess is that there is a higher success of fertilization with the, I can only speak to sort of pair spawning versus group spawning, and there's probably a higher success with pair because they're closer together, but also fewer eggs being released in a pair spawning. With respect to primary and secondary, I don't know that there would be a huge difference. I think it actually would rely more on the behavior and not necessarily how they got to be males, if that makes sense.
Okay. I think that is it, unless there are any other questions from the attendees. If not, I think I will turn it back over to Lisa. Thank you, Dr. MAdreani. It was a pleasure.
Thank you.
All right. This recording will be live on the Scas website, probably later tomorrow. Thank you all for coming and have a great night.
Thank you, everyone.